and I will be talking about them, and they're not going to be as toxic to our body and as toxic to our ethics as other solutions that have been. Now, first of all, we need to ask, is cellular regeneration and cell proliferation, is it the whole story? And the answer is no. Basically, like all the other speakers explained, is the differentiation of the cells that makes the difference. So what Xanya, for example, found and other investigators was that as you're aging, proliferation increases. So you have more cells, but you don't have differentiated cells. So differentiation goes down. Your proliferation goes up, differentiation goes down. In other words, you have more cells, but what do the cells do? They just sit there and they form tumors. And so the answer, again, is that differentiated cells. It's not just more cells, but the cells have to be differentiated. So the focus should be on differentiation. And the focus is how can we enhance differentiation of the cells. Now, there's a lot of anecdotal studies, like single studies, and there aren't any experimental studies. The problem with clinical studies is that you only get all your good responses, and the people that do not get that much difference you don't mention, or so basically you need a real experimental study so you know what's going on. So there was such a study started in 2005 in Johns Hopkins University, and they studied 48 human subjects, and they were looking at the safety of injecting adult stem cells at varying dosages. Now in 2008, they came back and they said, wait a minute, if we block potassium channels in these embryonic stem cells, then that slows down their growth and differentiation. So basically now we have a solution here. The focus can go on potassium channels, and we need to look at if these potassium channels are not blocked for any reason that they may be blocked, then differentiation of adult stem cells or embryonic stem cells can happen. Now, why is the differentiation necessary, and why is just simple proliferation a problem? Well, the reason is that, like the other speaker said, stem cells and cancer have a lot in common. They both have aberrant telomerase, and they both proliferate continuously. They don't have an end. So they have an infinite capacity to divide. Now, Gilbert is a London University professor and discusses all this evidence and how the stem cells are the driving force, and they're even the driving force in tumors. So even in cancer, the cells that cause the metastasis are actually cancer stem cells. And all cancers arise from cells that can divide. So in 2006, Michael Clark and Robert Weinberg postulated that in the heart of every tumor lie a handful of aberrant stem cells that maintain malignant tissue. So now what we're looking at was looking at another solution of how we can target stem cells in cancer and just try to destroy those instead of trying to destroy all the cells like chemotherapy does today. Now, here are the solutions. Number one, potassium channels. They're instrumental in stem cell differentiation. And they are obviously, that's a necessary thing to avoid tumors. So there is a book that came recently, and it was talking about electron truncated ion channels. And that will bring me into signaling technology is the technology that basically manipulates ion channels. It passes signals through ion channels. And what what the book postulated, and a lot of research obviously around it, was that the ion can control the gates of the cells, the ion channels, but it has to be at certain extremely low energies, which are below thermal noise. Now, signaling technology can amplify or substitute cellular signaling. It has to do with signaling pathways and ion channels. And aging and disease is seen as the result of deteriorated signals, specifically protein signals, by a number of investigators. And cancer is, from this point of view, is seen not just the result of a number of rebel cells, like we thought about cancer before, that refuse to follow the rules, and therefore that refuse to fulfill all the steps of regeneration or mitosis very much. So they just go on and they keep proliferating. But it really is seen as a group of cells that they are obedient, however they have gotten the wrong directions, they got the wrong signals. Now in 2011, there has been some proof that cancer is the result of cells getting the wrong signals, that they're giving the wrong instructions to them. And how can they get the wrong instructions? From the very beginning, from the DNA that is duplicated by the RNA, there can be something wrong there due to radical damage, free radicals that damage the DNA or other factors. So now you can also, the natural proteins, for example, can cause problems. Just a simple substitution of one letter in the DNA can be the cause of cancer. Now, the 2005 to 2011, scientists, we are talking about the WNT signaling pathway. Now, the WNT signaling pathway can act to maintain stem cells, as well as cancer cells, of course. And the ability to modulate that WNT pathway, either positively or negatively, because you can turn it off and then turn off the proliferation completely, that would be of therapeutic relevance. So now we have a second solution, the WNT pathway, along with the potassium channels. And again, these are at the cellular level. So utilizing signal technology, you can control the activation through signals, basically, that can stimulate the activation of the WNT signaling pathway. And inhibition of that pathway, the WNT signaling pathway, can prove to be an effective route to inhibiting the uncontrollable renewal that drives the cancer. Now, another quite famous, actually, agent of stem cells is the p53 gene that causes the cell cycle arrest. It basically stops any division and it starts cell death. But it's a more risky one because we don't have the exact signals that can stimulate that. We have only approximate signals, and therefore it could cause more damage. But it's a semi-solution, if you like. Now, Howard Hughes Medical Institute, Elaine Foots, has looked at the BMP pathway. It's another signaling pathway along with the WN2 pathway that can influence stem cells, and they can coax them to differentiate. So that's again signaling that can cause a stimulation of the BMP pathway, can enhance the cellular differentiation. Another pathway is the HMGB1 signaling pathway. And by tapping this signaling mechanism, researchers who develop new treatments for skin injuries like severe burns. Now, what that is, it's like a speakerphone, like a megaphone in the system, that what it does, it gives out a distress signal, so the cells can come to the area of the wound or of the trauma. So isolating that signal of the WNGMP1 can solve a lot of very serious medical conditions, and that's what researchers are reporting from London University, where I'm familiar with, and also Suffolk University in Tokyo, in Japan. Now, in 2011, we also looked at the HB1.F3 cells that can induce neuronal differentiation of the neural cells. Now, solutions in problems arising with stem cell therapy so far are based on inducing stem cell differentiation by targeting potassium channels, WNT pathway, the BMP pathway, the P3 gene activation, which is again not such a great solution, but it should be there, the neuroT factor, and the HMGB1, that is like a megaphone for the system, that can basically get the stem cells it can attract the stem cells, or it can tell the stem cells where to go. So utilizing signaling technology to decode different biological signals and then reproduce them can help us with producing these solutions. And this is just how some of these waveforms are composed. It's basically digitally, and then you sort of target cells, but you have to turn them into uneven resolution first, otherwise the cell will not respond to the digital signal. This is some more signals that we created when we were working on stem cells. Some more of different signals. You can see there's different waveform combinations, and they're extremely low current. It's about 5 or 10 nanoamps, which is below thermal noise. And the idea is basically you need to have, it needs to be that low, or it's not going to control the ion channels. The ion will not control the ion channels. And thank you very much. This is.
Thank you for your interesting talk. Are there any questions on the floor? I have a question. Most scientists believe in Knudsen's multiple hit hypothesis about cancer formation. What you are proposing is very, I would say, radically different. What do you think about Knudsen's multiple hit hypothesis in cancer formation?
Oh, I think this is obviously what is the truth. I mean, the truth can come from different ways. This is just one part of the story. It's just one aspect of it. And it's an aspect that you look just at the signal, but there's just so many more aspects of this disease that we have to look at. So this is just one aspect, but of course, it's just a small part of the truth, let's put it that way. It's just a whole different perspective.
Are there any other questions from the floor?
How would you think that these signaling pathways like BMP or potassium channel would be practical in the guide for developing future stem cell research? We know from basic research that all these signal is part of the microenvironmental niche where the cell used to determine its fate in development and differentiate. In many areas, it's almost like textbook knowledge now. Like in developmental neurobiology, we teach students all these signaling pathways and how the cell will be induced from the neuroepithelium to various types of cells when they migrate from the gland. But you mentioned 2 points that when, for example, calcium channels are altered and the cell, the stem cell fail, further, it would be converted to cancer. How is that?
The research that came from Johns Hopkins, primarily, that's what I was talking about, is that if they block the potassium channels within the stem cells, then the stem cells will not differentiate. You see,
which kind of potassium channel? There are many types of potassium channels in cells, like heart muscle cells. There are the voltage-sensitive channels,
all potassium channels,
and also the passive and the lead channel.
It's all potassium channels. It's just specific only to potassium. Any potassium channel there. In cellular research, it's much simpler than when you look at more, organs, more combined things. I think the answer to your question is that we will have to depend on more technological developments because we need better microscopes, we need better computers to kind of analyze all of these signals. We need better digital to analog converters to basically translate these signals into something that is more. So we depend on technology, and as a matter of fact, it wasn't really until 2011 that we were able to even kind of believe that we can get these signals. We wouldn't be able to get them before that.
The classic textbook said that the potassium channel is responsible for the resting membrane potential that create the difference between inside and outside the cell, right, which maintain, and without that, it affects many other cellular processes.
Yeah. Well, a lot of stem cells, that's all they have. They don't have calcium channels, they just have potassium channels. So that is the problem. They just have only one type of channel there. So because they lack calcium channels, I think the potassium channels becomes very crucial. And I think that's why it becomes such a, they're just blocked. And it's like a house with no windows and no doors. So nothing can go out. There's no signals passing. So obviously the cell cannot get, not only, it's not that it doesn't get the correct signals, it probably gets no signals whatsoever. So of course it's going to turn into cancer and not differentiate. And that's sort of a more logical kind of answer to your question.
Are there any more questions on the floor? If not, thank you, Dr. Xanya Weiss.
Thank you for having me. Thank you very much.
This concludes the